Welcome for this afternoon session. Thanks for the kind introduction, Vladka. And I will talk on two subjects, actually both on glycosphingolipids in cancer. The first one is done with our in-house collaborators, Jing and Peter, and the other one was done entirely by our own group on glycosphingolipid analysis in cancer, as I said. Good. So what we did is analyze glycosphingolipids of cancer cell lines. Now, what I will present is kind of related to what Guinevere presented. Her presentation was on tissue analysis, colorectal cancer tissues, analyzing the O glycans and the N glycans there. We were not able to do glycosphingolipids there, in part because those will be washed away during FFPE embedding. So for glycosphingolipids, which are implicated in many cancers, we relied on cell lines to analyze their role in cancer. And these are two examples of colorectal cancer cell lines which we typed. And if you look at these patterns, we have on top a colon-like cell line and here a differentiated cell line. So cell lines are different and they have their benefits and in glycomic analysis or in biological analysis in general. So if you look at the top cell line, then you will see a lot of Lewis-type terminal structures, Lewis X, Lewis Y, all silent Lewis X, and so on. Whilst the undifferentiated cell line here has terminal GalNAcs, so that would be A-type blood group antigens. So blood group for undifferentiated, and then here for differentiated cell lines we have the Lewis-type structures. And these are not only two examples. What we are looking at are head groups of glycosphingolipids. I won't show the workflow in detail, but it's very much related to what Guinevere showed this morning. So we prepare the analytes, the glycosphingolipids. We use an enzyme to cut the glycan portion off. We then do the PGC separation in the very same way as Guinevere presented for O glycans and N glycans. And this is already kind of a summary of the first part of the talk, where I show that there's a differential expression of glycosphingolipids for undifferentiated versus colon-like differentiated cell lines. And here again, these structures are shown. So blood group type structures like these ones here with a B or A blood group antigen are in the undifferentiated cells, while with the differentiation of the colon of the epithelium, we see a upregulation of Lewis-type structures. Of course, we did not only do two cell lines, but some more, if I go in the right direction. And that's shown here in a PCA plot. In red, we see the colon-like cell lines. You see we did some 20 cell lines, or but some more. And then the undifferentiated ones are down here in the right bottom corner. And if we look now in the loading plot at which glycans are driving these separations, this very nice separation, I want to say, then we see indeed that upon differentiation, these cell lines, or all the differentiated ones, have the Lewis-type structures, fucosylation, and so on. Neolactoserous glycosphingolipids, whilst gangliosides are down here, and also blood group type structures, as I mentioned, B, A. So that is, of course, reflecting the blood group of the patient from which the cell line has been derived. So this is the summary slide here in terms of the differential expression of glycosphingolipids. And now, the title of my talk referred to the integration of transcriptomics and glycomics data, and we find this very insightful, not per se to prove how biosynthesis happens, but to reveal some nice indications. What we did here is we profiled the glycomes or glycosphingolipid glycomes of these cell lines. We took transcriptomics data which were publicly available, and we looked at associations. We did that on the one hand for the glycosyl transferases from these public data, but also for transcription factors, which are implicated in the differentiation of the epithelium of the colon. And what we see here is quite strong associations of the glycans with specific transcription factors. Here we have MYB, we have HN1 alpha, HNF4 alpha, we have CDX1. These are all well-known transcription factors driving the differentiation of the epithelium. And I must say I was astonished to see that we have a very strong association of these factors with the glycosylation features. And on the other hand, I have to say that the association of the glycosyltransferases is often somewhat weaker. And this is still kind of a mysterium. We find this more often, that the transcription factors are dictating the glycans, whilst the transferases, which are directly implicated in the synthesis, have a less strong association. Yeah. Then. So this is kind of what we derived from this is now just touching back onto the O glycans, because until now I talked about the glycolipid head groups, and now in the same cell lines we also have O glycans, which we released the very same method which Gwyneth presented this morning. And also here we see a gradient of expression of glycan structures for these O glycans. We see the very same transcription factors implicated or associating with the expression of structures. And again, we see a expression of Lewis-type structures in the codon-like cell lines. So some similarities in the head group expression for the O glycans and for the glycolipids. And then, so this was already the first part of my talk, which was on colorectal cancer cell lines and their expression of O glycans very shortly, but then also of the glycolipid head group structures. Now, what I want to talk about in this second part of my presentation is two cell lines, which is a murine breast cancer cell line and a human lung cancer cell line. And what we did in this case is more functional studies together with our in-house collaborators. Now, if you compare the Glycome profiles we have here with the ones I just shown you, we see a very big difference, and that is indicated here because these cancerous cell lines express A-series gangliosides. So for all the colon-type cell lines, we were in this region here, expression of neolactoseries glycosphingolipids. So that's colon epithelium. What we now see for these breast cancer cell lines is ganglioside synthesis, and then you have the ST3-GAL5 synthase, which puts the salicylic acid here onto the galactose. And what we see here is that all these glycans here, these gangliosides, are highly expressed in these cell lines. And now our collaborators is Peter den Dijke. He is interested in signaling in various cell settings, in cancer settings, and what he studied is now adding TGF-beta as a mediator to these cell lines, to the culture, and looking how does the glycosylation of these cells change. And we looked at N glycans, O glycans. I will not show you that to you, but we saw, interestingly, that the profile of gangliosides did not change at all. But luckily, we included an internal standard for a quantification, and that allowed us to conclude that actually all of these glycosphingolipids were going down, all these A-series gangliosides were going down upon TGF-beta treatment. We saw that both for the murine cell line and for the human lung cancer cell line, a decrease of the expression of glycosphingolipids. And now I will zoom out into a model. So we did many more studies on these cell lines, and what we found out is actually that indeed the data I showed you is now on this left-hand side. TGF-beta signaling results in a lower expression of glycosphingolipids and less expression of gangliosides, also lower expression of ST3-GAL5. What we then wondered is how do glycosphingolipids, on the other hand, impact the signaling. So this is the signaling impacts glycosphingolipids. Now the question is, how do glycolipids impact TGF-beta signaling? And what we found, to tell you that already now, is increased levels of GM3 result in a degradation of TGF-beta receptors. And we will now spend a few slides on this intriguing aspect. We used now a bunch of technologies to unravel these associations. We looked at knockdowns, or knockouts, I should say, so no more glycosphingolipid biosynthesis. We also used a small molecule inhibitor, which inhibits the synthesis of the whole glycosphingolipids, also of the gangliosides. We used ultracentrifugation of the membrane fractions to separate these microdomains. So we have a raft microdomain where TGF-beta receptor is normally located, and with the glycosphingolipids added, the non-raft part, and with the glycosphingolipids added or increased, the TGF-beta receptor gets into these rafts and gets degraded. And here are some data now in animal models, also functional data. So what we showed here, or what our collaborators showed, is overexpression of SD3-Gal5 on these cell lines results in less extravasation of the cancerous cells. So in this model, you look at functionally at these cancer cell lines. In this case, they have low glycosphingolipid expression, and the cells are invasive and get into the tissue, and you see the red dots here. In this case, we have high ganglioside expression, and the extravasation, the migration of these cells into the tissue here, is prohibited. This is then also quantified here, and indeed you see with SD3-Gal5 overexpression, less extravasation of these cancer cells. Now the next part here, these slides here are on the human side, again on lung cancer. What we see here is if you look at the expression of SD3-Gal5 in lung cancer, in the tissue, the high expression comes with a better survival of the patients. So ganglioside expression, a series ganglioside expression, protects the patients to a certain extent, and it also helps in the zebrafish model to avoid metastasis or invasion of these cancer cells. Then if we look in the tissue, we see indeed that the cancer tissue has a lower expression of SD3-Gal5 compared to the compared to the healthy lung tissue, adjacent tissue. And this is quantified here, so indeed, in all these cases, in a paired comparison, the adjacent lung has a higher expression of gangliosides than the cancer. So low ganglioside expression is bad in lung cancer. That is kind of a simple summary here. We also moved into animal models here for mouse models. It was not that clear, the data. But in the end, what we showed in the mouse model is, to a certain extent, inhibition of ganglioside synthesis in these cancer cells led to more tumor growth. So again, a bad outcome if you inhibit a series ganglioside synthesis. And this is the mouse situation is paralleled again by the zebrafish situation. So what I showed before is a overexpression of gangliosides by overexpression of SD3-Gal5. Now we inhibit ganglioside synthesis, and then indeed it goes in the other direction. This inhibitor here comes with more extravasation, so more cancer, and you can inhibit TGF-beta signaling, and by that you can reverse this effect again. So what we can conclude is somehow these gangliosides, which are the gangliosides are reduced with TGF-beta signaling, but they also themselves again affect TGF-beta signaling. So it's kind of a close interplay between gangliosides and TGF-beta signaling. And that is what I try to summarize here on this slide. So indeed, the left-hand side of the scheme shows TGF-beta signaling leads to a lower expression of gene 3 of the A-series gangliosides. On the other hand, gangliosides themselves influence TGF-beta receptor degradation. So we have a whole bunch of data in this manuscript which support that. I have no time to show that here. But actually, this brings me already to the conclusions of my talk. So regarding the colorectal cancer, what I showed you is that Lewis and blood group antigens are differentially expressed in colorectal cancer cell lines. And so they reflect the differentiation status of the CRC cell lines. Now, if we look now at the TGF-beta signaling, A-series ganglioside expression in lung cancer inhibits TGF-beta signaling and comes with a good prognosis. So just to rephrase this, so the A series gangliosides are a positive factor in lung cancer. And that means in bad prognosis, bad outcome of patients, those bad tumors have low expression of the gangliosides. And on the other hand, TGF-beta treatment of lung cancer cell lines decreases A series ganglioside expression. And what I want to stress here is, for me, this was kind of an eye-opener. So what comes first? What is first? Is it the signaling or is it the glycosylation change? What we see is TGF-beta signaling leads to a change in glycosylation, but also a change in glycosylation, again, leads to affects TGF-beta signaling. So in the end, I think it's not going to be possible to say what comes first. These things come hand in hand, and they mutually support each other. So in that respect, the question, is glycosylation change a cause or a consequence of cancer or cancerous processes? I would say it's an integral part of malignant processes, and it's, on the one hand, lower gangliosides are driving this cellular degeneration, but also cellular degeneration is driving glycosphingolipid changes. Glycan changes, cellular phenotype changes, they come hand in hand, and this is mediated by this TGF-beta signaling. Okay, this is then my last slide to thank you and thank my collaborators and my colleagues. This was mainly work done by Dee, Katharina, Tao, Oleg, and Guinevere, who is here in the audience, and then our collaborators Peter and Ying from the cell biology department. Thanks. Now, I'm also chairing the session. Let's have any questions at the end, I would suggest. But I think I will need help here with the slides. So the next presentation is by Yuri Oljenko. Hi, Yuri. You ready?